Hi everyone, welcome to Teacher Gideon Learning Channel. And if you have joined me for the first time, make sure you subscribe, you like, you comment, and share. Today I want us to learn about grade 8 work, specifically under the strand of living things and their environment. And before we start analyzing about the cell, you find that living thing is just a living organism, and this living organism is being surrounded by both biotic and abiotic factors. So the surrounding of an organism is known as the environment. So the cell that I want us to learn about is the cell, where we are going to define the cell. We are also going to learn about the structures found within a cell, differences between a plant cell and animal cell. We are also going to learn about functions of different structures. We're going to compare a plant cell and animal cell. So the cell make up the structure of living organisms and carry out various biological processes. Organisms such as amoeba are composed of single cell, hence are said to be unicellular. Organisms such as plants and animals are composed of many cells, hence are said to be multicellular. So you are supposed to know that if an organism is comprised of one cell, it is known as unicellular. But if an organism is comprised of more than one cell, it is known as multicellular, meaning that it's having many cells. Mult means many, uni means one. So, organisms such as amoeba are composed of single cell, hence are said to be unicellular. Organisms such as plants and animals are composed of many cells, hence are said to be multicellular. Therefore, a cell is defined as the basic unit of structure and functions of the organism. So that's the meaning of a cell. A cell is the basic unit of the structure and functions in organisms. To observe the cell, a powerful magnifying instrument called a microscope is used. A microscope enlarges the image of objects when observed and improves the resolution of the image. Remember last time we learned about undergrad 7 work, I did a video explaining about the microscope. So under that video, we learned about what's a microscope, what's the function of the microscope, what are some of the parts of the light microscope and their functions. And also remember we learned about two types of microscope. We have the light microscope, electron microscope. We also analyzed how you are supposed to care and handle the light microscope. So the microscope is being used for enlarging an object or to magnify an object. To magnify is to enlarge something. So if you want to see finer details of the cells, you are supposed to use a microscope. Then plant and animal cell structures are seen under the light microscope. The following diagram shows the components of the plant cell as seen under the light microscope. So you're supposed to know the parts of the plant cell, structures that are being found within the plant cell. The outermost one is the cell wall. We are going to analyze also the functions of the cell wall. Then the second layer now is the cell membrane. Cell membrane. Then after the cell membrane, you find that there is an arrangement of chloroplasts. There is a cytoplasm. There is a vacuole. This is the vacuole. Then there is the nucleus. Then there is the nucleus. So those are the parts of the plant cell. Then we also have the following diagram shows our different components of the animal cell seen under the light microscope. So when you use the light microscope, you are going to see the parts that are being found in animal cell appearing like this diagram. So we have the cell membrane, nucleus, and cytoplasm. So from those two diagrams that we have seen, both the animal cell and the plant cell, we can be in a position to compare or give the differences between the plant cell and the animal cell. Plant and animal cells have slight differences in their internal structures, while some features are common in both. The figures below shows the components of the plant cell and animal cell as seen under the light microscope. So from under the observations, you can be able to make the differences between a plant cell and animal cell. If you are keen enough and you look at the first diagram of the animal cell, there is no nucleus. There is no cell wall. But when it comes to this plant cell, there is a cell wall. Also, you can be able to see at the first diagram, there is no chloroplast. But at the plant cell, we have the chloroplast. Also, from the first diagram, you can be able to see we don't have a vacuole. But under the plant cell, we have the vacuole. So let us analyze the differences. Plant cell, they are large in size. Then when it comes to animal cells, they are small compared to plant cells. So you find that these animal cells are very small compared to plant cell. So one of the differences you're supposed to compare based on the size. Plant cell is large in size, animal cell is small in size. Then plant cell, it has a cell wall. Then animal cell does not have a cell wall. Does not have a cell wall. Then another difference, some plant cells have chloroplasts that give the plants its green color. Then animal cells lacks the chloroplast. So you find that under plant cell, the chloroplast is the one that is containing the chlorophyll. Inside the chloroplast, we have the chlorophyll. And the chlorophyll is the green coloring matter. Chlorophyll is the green coloring matter. It's the one that makes the plant cell to be green in color. And the function of this chloroplast, it is a site for photosynthesis. Remember, photosynthesis is that process by which a plant makes its own food using the light energy. Then when it comes to animal cells, it lacks chloroplast. So there is no chloroplast. And remember, if there is no chloroplast, there is no chlorophyll. And this makes animals not to be green. So animals can't make their own food because they lack the chloroplast. Then another difference, plant cells have a permanent vacuole that occupies a large part of the cell. Then when it comes to animal cells, usually do not have vacuoles. However, small temporary vacuoles may occur. So most of the animal cells, they don't have a permanent vacuole, but sometimes you may have the small one. I mean, very small and temporarily, just for a short period of time. Then similarities between a plant cell and animal cell. They both have a cell membrane, both have the nucleus within them. Next, let us analyze about the functions of the structures found in plants and animal cells. Before we analyze about the functions of those structures found between the plant cell and animal cell, you're supposed to know these ones: the vacuole, the cell membrane, the cytoplasm, nucleus, cell wall, chloroplast, are known as organelles. They are being known as organelles. They are being known as organelles. And these organelles are just structures within a cell. So you find that organelles are just structures that are being found within a cell. And when these many organelles combine together, they form a cell. And when many cells combine together, they form a tissue. And when many tissues combine together, they form an organ. And when many organs combine together, they form organ system. Now, when many organ systems are being combined together, they are going to form a living organism. So that is what you are supposed to know. So organelles are just structures within the cell. And I've said, let me try to draw that structure. Organelles, they form a cell. Cell forms tissues. Tissue, they are going to form an organ. Organ is going to form organ system. An organ system is going to form living organism. So you're supposed to know that the flow, where you find that organelles form a cell. Many cells, when combined together, they form a tissue. And when many tissues combine together, they form an organ. And when many organs combine together, they form the organ system. And when many organ systems are combined together, they form a living organism. So we have functions of those different parts or structures found within the cell. We have the vacuole. It is the space that has watery fluid, which is known as the cell sap, that contains dissolved water, mineral salts, and waste products. So you find that under the vacuole is just watery, which is having the type of that liquid that is being found inside the vacuole is known as the cell sap. And this cell sap is being dissolved with water, mineral salts, and waste products. So you are supposed to know under the vacuole, it is just being used to store the mineral salts, food, water, and even some waste products. So the name of that vacuole that is being used to store food is known as the food vacuole. Food vacuole. Then the one that is being used to remove the waste products is known as contractile. Is known as contractile. Let me write here. Contractile vacuole used to remove the waste products from the cell. So vacuole are being found both plant and animal cell. But make sure you know that if it's being found in the animal cell, it is very small in size and just temporary. Then cell membrane. Cell membrane is a thin layer around the cell that holds the cell together. It acts like a fence and controls what goes in and out of the cell. Therefore, the cell can take in substances it needs and get rid of waste products. So the function of the cell membrane is to allow substances in and out of the cell. But you're supposed to know this cell membrane is semi-permeable. Semi-permeable, where you find that it allows some of the substances to enter and preventing the others from entering. Then you find that this cell membrane is being found in both plants and animal cells. We have the cytoplasm. It is a jelly-like liquid that fills inside the cell. The cytoplasm is where chemical reactions of the cells take place. So the function of the cytoplasm It is a site for chemical reactions. It also contains small structures called organelles, which have special functions. So you find that this cytoplasm is being found in both plants and animal cells. And remember, cytoplasm is a site for chemical reactions. Another one is the nucleus. The nucleus carries the genetic information and controls what happens or all the activities of the cell. So you find that this nucleus, the one that carries the genetic information, or you can just say the DNA, carries the DNA. DNA in full means deoxyribonucleic acid. Deoxyribonucleic acid. And this nucleus also controls all the activities of the cell. And the nucleus is being found in both plants and animal cells. Then we have the cell wall. It is a thick, tough layer made from cellulose found on the outside of the cell. It covers the cell membrane and plant cells. Cell wall help the cell to keep its shape, protects the cell from mechanical damages. So one of the functions of the cell is to give the plant cell the shape. Remember, this cell wall is being found in the plant only. So it gives the plant the shape. Then also it protects the cell from mechanical damage. Protect the cell from mechanical damage. It is being found in plant cell. Then we have the chloroplast. It's an organelle in the cytoplasm of plant cells. Chloroplasts chloroplasts contain green pigment called chlorophyll. Chlorophyll absorbs the light that is used by plants to make their own food through photosynthesis. So you find that this chloroplast is the one that contains the green pigment known as the chlorophyll. And this chlorophyll is the one that absorbs the light energy from the sun, enabling the plant to make its own food through the process known as photosynthesis. Then the last part of this strand is about magnification. Magnification of cells. Magnification of the specimen is the measure of how much bigger a specimen is when it is viewed through the hand lens or microscope compared to its original size. Magnification is usually expressed using times. Don't say x, it is times. Before the digits, for example, times 2, times 10, times 20. The x stands for times. Yeah. If it's times 2, it means that that specimen has been magnified 2 times. It is a measure of how much bigger an object appears when viewed through a hand lens or microscope. For example, times 2 means the object has been magnified or enlarged 2 times, so the image is twice bigger than the actual object. For a light microscope, the final magnified image of an object as seen by the observer is the product of the magnifying power of the lenses that are used. These lenses include eyepiece lens and objective lens. The magnifying power of each of these lenses is marked on the sides of the objective and eyepiece lens holder. In a light microscope, the revolving nosepiece holds these 3 different objective lenses, each with specific magnifying power times 10, times 4, times 40. The eyepiece lenses further magnifies the image formed by objective lens. Calculating the total magnification. So for you to calculate the total magnification using the light microscope, just take this formula. Magnification of the eyepiece lens multiply by the magnification of the objective lens. Then what you get, you must put times. If you get times 50, write times 50. This one shows that that organism, that specimen has been enlarged 50 times. There is an example below And I want you to take the screenshot of that example. You fill in the table below with the correct magnification. And remember the formula is magnification of the eyepiece lens multiplied by the magnification of the objective lens. So I wish you all the best. Thank you for having my time, and I appreciate continuing in the same spirit. Bye bye.